Welcome to Sense of School of Life Sciences. Today's topic of our discussion is Transposons Part 1. In this video, mainly we focus on the major classes of transposons. Okay, transposons or jumping genes are short DNA sequences that have the ability to move from one location to another in genome by the process of transposition. The DNA sequences that is transposed may be selfish DNA, moderately repeated DNA, etc. Selfish DNA, for example, it has no selective advantage for the organism, isn't it? The repeated sequence may be moderately repeated or highly repeated sequence. Moderately repeated sequence includes signs and lines, which are retrotransposons that we will study in separate class. Other DNA sequences that is transposed that we will study now. Then what is safe havens? Transposons can control target site choice. That is, transposon can control target site where the transposable element is inserted. Two general types of target site regulation are involved. In the first, some elements preferentially insert into regions of the chromosome that tend not to be harmful to the host cell. That is when the transposon is inserted into another location that may not be harmful to the host. These regions are called as safe havens for transposons. Then, in the second type of regulation, I told you two types of target site regulation are there. In the second type of regulation, some transposons specifically avoid transposing into their own DNA. This phenomenon is called a transposition targeting immunity. Okay. Safe havens and transposition target immunity. These two words also remember, sometimes we asked. It was Barbara McClintock in 1940s who discovered transposable elements in maize genes that caused multicolored kernel. She identified that the controlling elements AC and DS are responsible for genetic instability of corn seed color. So coming to the classification, we can broadly classify transposons into two major classes as DNA transposons and retrotransposons. DNA transposons are also known as class 2 elements, and retrotransposons are known as class 1 elements. The major differences between these two. See, in DNA transposon, transposition occurs through DNA intermediate, while in retrotransposons, transposition it is through RNA intermediate. So there is the involvement of reverse transcriptase enzyme, and it is a type of copy-paste mechanism of transposition that we'll study later, because today we are mainly focusing on DNA transposons. See, about the DNA transposons, it has terminal inverted repeats. You see, there's a terminal inverted repeats. Here ATTCGGCA, here ACGGCTTA. It is inverted segments. A gene encoding the transposase that is responsible for transposition. That is transposase is there. It is the simplest one. We'll say IS element in bacteria. It's an example for DNA transposons. It's the simplest one. So once more, here DNA transposons, also known as class 2 elements. Here transposition through DNA intermediate carries terminal inverted repeats and a gene encoding transposase that is responsible for transposition. And these examples also is very important. IS elements, it is the simplest. It is seen in bacteria. P element in Drosophila is very important, especially hybrid dysgenesis in Drosophila that we will study in separate classes. Very, very important. Then ACDS maize in maize, TN3, TN10, etc. and in E. coli, TAM3, Snapdragon, bacteriophage mu. So all these are familiar examples. Then I told you retrotransposons in detail we'll study later, but you just see the classification. Retrotransposons can be broadly divided into two as LTR retrotransposons and non-LTR retrotransposons. LTR retrotransposons, also known as retroviral-like retrotransposons. It is not retrovirus retrotransposome, it is retroviral retrotransposomes. And non-LTR retrotransposome known as non-retroviral retrotransposomes. Here also the examples are very, very important. The example Copia in Drosophila, Ty1 in yeast, THE1 in human based on maize. Similarly, in the non-LTR retrotransposomes, example F element in Drosophila, SYN4 in maize. In human, lines and signs. Lines and signs, I told you, it's a repetitive DNA sequence at the same time as a retrotransposome. Okay, so remember this classification very well, because it's a very important this classification. So in which category each transposome comes is very, very important. Then now we can focus on DNA transposomes for class 2 elements. There are two mechanisms of transposition in DNA transposome. This is one cut and paste mechanism, other one copy paste mechanism. In cut and paste mechanism, there is cutting of DNA that is considered as the transposome. You see, suppose the DNA, DNA, we can consider this as the transposome, and then it is pasted in a target site. Let it be, this is the target site where it is pasted. So actually it's a cut and paste and no copy is formed, cut and paste. So when the DNA is cut here, it leaves behind a hole in the chromosome here. This will create a hole in the chromosome here. Then this lesion can be healed by recombinational double-stranded break repair. Okay. In any way, when it is pasted, that will be like this. Then, moreover, it is known as conservative type. The cut and paste mechanism, it is known as conservative type. But why? It is the same DNA which is pasted here. It is the same DNA that is conserved through this mechanism. Okay, it is conserved type or conservative type. Don't forget it. It's very important. And no change in copy number. There's only one copy is there. Okay, no change in copy number. Then what about the copy paste? It is known as replicative type. What do you call replicative type? Here, a copy of DNA is formed by replication. It is pasted here. Okay, so a copy is formed and it is pasted in the target site. So here, increase in copy number will be there. Here only one copy and two copy. So increase in copy number is there. So cut and paste mechanism, it is conservative type and no change in copy number. And copy paste mechanism, in copy paste mechanism, it is increase in copy number, it is replicative type. See, just to remember, copy paste mechanism is different in retrotransposons. There I mentioned, there we can see the copy paste mechanism, but it is different type. There it is through RNA intermediate. It is very important that we'll study in retrotransposome class. Okay, then we can study some facts about the different types of DNA transposome. DNA transposome, once again, you see the example IS element bacteria, P element ACDS means in TN3, TN10, etc. TAM3, Snapdragon, bacteriophage meal. So we shall mention some points of this on these IS elements, then bacteriophage, then some of this DNA transposons. So the bacteriophage, you see, bacteriophage the bacteriophage not only uses DNA-based transposition to integrate its genome into the host, it also uses replicative transposition. It is replicative transposition to replicate its genome. It's a very large bacteriophage transposome is a very large transposome. You remember that bacteriophage transposomes can be divided into three types. What are insertion sequence, composite transposome, non-composite transposome, like this. So what are insertion sequence or IS element, composite transposome, non-composite transposome. IS element, already I mentioned, it is the simplest one. This diagram shown there. IS element is the simplest form of transposome. The IS element contains nothing but one gene only. What is transposase gene? The transposase gene, actually, it will catalyze the movement of transposon to another part, nothing else. So the terminal inverted repeats and direct repeats are also present. Direct repeats also is present, just outside. The inverted repeat is this. This is direct repeat. No other genes are present inside. Simply cut it and paste it. So here, cut and paste mechanism is there. It gives no benefit to the bacteria. Then I told you three types: insertion sequence, and I said it is then composite transposome. Composite transposome. Composite transposome. What is the peculiarity? But between the two such elements, if contains genes which carries antibiotic resistance feature, the bacteria will get benefit. This type of transposable element, which contains apart from transposase gene, is known as composite transposome. Composite transposome, what is the main peculiarity? You see, this is IS, this is IS. Two IS elements are there. This is one peculiarity. And antibiotic resistant gene is there. Here, IS10, why? It is TN10 bacterial transposome. Here, the figure, this diagram is that of what TN10 bacterial transposome, where the antibiotic resistant gene is tetracycline. Remember it. Composite transposome example TN5, TN9, TN10, etc. Here the diagram it is TN10. That is why here with IS10. So in composite transposome, once again, I tell you, unlike the IS element, which is simplest, here there is antibiotic resistant gene. And moreover, IS elements, two IS elements on either side can be seen. Okay, the example, very important, that the composite transposome will have two insertion sequence, very important. Then, how it differ in non-composite transposome? In non-composite transposomes, there is no IS element, very important. But the terminal part is not IS, but the terminal part is inverted repeat. Only the inverted repeat will be there. But like the composite transposome, antibiotic resistant gene will be there. Okay. So in one way, it resembles the composite transposome. But in what way? In which way it differ from it? It has no IS element on either side, only inverted repeat. Okay. An example important TN3, TN7, etc. Non-composite transposomes are lengthier than composite transposome. Okay. Then what is ACDS element in this? You know, it was discovered in maize. ACDS transposable elements was the first transposable element system recognized in maize. McClintock found that the kernel mosaics in maize is frequently resulted from breaks at a particular site on chromosome 9. In the presence of activator AC element, a dissociation element can cause chromosome breaks. AC element is autonomous. But one thing remember, so you understand, breakage of DS loci in chromosome 9 occur only with the help of AC. So AC is autonomous element, whereas DS element is non-autonomous because it cannot produce transposes needed for transposition. So DS element is dependent on AC element. Details of AC DS element we shall study in separate class. Then about the autonomous, non-autonomous. Autonomous and non-autonomous transposes are also seen in retrotransposons. For example, lines are autonomous and signs are non-autonomous. That we shall study in retrotransposon class. Okay. Then we can do some question. First question. DNA transposons move directly as DNA sequences and use transposes to cut and paste into the gene. Which of the following is a well-known transposon in maize corn? Here, the first part of the question is only a general statement. The question is, which of the following is a well-known transposon in maize? It's a direct question. It is what ACDS element. Which of the following is a non-composite transposon? It's also a direct question, TN3. Okay. Then, transposons are made of segments of RNA, segments of DNA, antibody, protein. What is segments of DNA? It's an answer. A transposon can do all of the following except A, move from a plasmid to the recipient cell genome. B, lyse the host chromosome to which it is transferred. Move from one site on the host genome to another site on the host genome. Move from a host genome to a plasmid. Make a copy of itself, leaving the copy in the original position as it moves. You see, all the are correct, but the second one is lyse the host chromosome to which it is transferred. No, it does not. It does not lyse the host chromosome, isn't it? So it move from a plasmid into the recipient cell genome. Is move from one site on the host genome to another site on the host genome. All these are correct, and only B is not correct. Similarly, transposons also can move from plasmid to bacterial chromosome and vice versa. Okay, let also remember. Then more question. When a transposon moves within a host cell, it undergoes transformation. Always removes all traces of itself from the original location. No, not always. Sometimes leaves a copy of itself at the original location. It's okay, it's correct. So C is the answer. Okay, if you like this video, please subscribe, like, share. Thank you.